Imagine this. You're a soldier in an army going into battle. Your leader knows the enemy's plan and has come up with a plan of attack. Thanks to advanced technology, he has upgraded your weapons to destroy the enemy based on their plan. By the end of the battle, the enemy has no choice but to surrender. Now what if I told you that this planned attack method is the next step towards curing cancer? Hi, I'm Danielle Kui, and today I'll be talking about CAR T-cell therapy, a revolutionary cancer treatment primarily used to treat blood cancers. Before we can talk about this treatment, we need to understand what the immune system is and how it works. The immune system consists of many cells and tissues that work together to keep us healthy, creating an arsenal of weapons prepared to fight any microbes that invade the body. One of the key players of the immune system is the T-cell. On the surface of these cells are receptors that are able to attach to antigens, protein fragments found on the surface of pathogens. When the receptor attaches to its corresponding antigen, the T-cell releases a signaling molecule known as a cytokine, initiating an immune response that ultimately ends in the death of the pathogen. Some of the T-cells remain long-term, so that when the antigen returns, the immune response will occur quicker. With cancer, we see a different story. Cancer cells present themselves with neoantigens, or antigens that the body hasn't seen yet. The T-cells can identify some of these neoantigens, signaling an immune response. However, some of them are overlooked, allowing the cancer to spread further. This is where the attack plan analogy comes in. Using what is known about the neoantigens on the remaining cells, researchers can genetically modify the immune system's weapons, the T-cells, to target the cancer cells. This is called chimeric antigen receptor T-cell therapy, or CAR T-cell therapy. Well, how does this work? First, blood is drawn from the patient. Then the T-cells are separated from the rest of the blood, which is returned to the body. Special receptors that target the neoantigens, known as chimeric antigen receptors, are attached to the surface of the extracted cells, which are then grown in a special lab. As I mentioned earlier, the immune system targets foreign cells, and CAR T-cells are no exception. Thus, before being given the cells, patients go through a short course of chemotherapy to increase the chances that the CAR T-cells won't be attacked. Then the cells are administered through an infusion. CAR T is slightly limited because it is autologous or patient-specific. This makes preparing the treatment labor-intensive, time-consuming, and costly. Thus, to counteract these drawbacks, off-the-shelf or allogeneic CAR T is being tested. How might this work? There are two methods that can be used. One, by taking T-cells from healthy donors, or two, by using pluripotent stem cells. The first method follows the same process as using the patient cells, the only difference being the source. The second method, however, is more complex. Let's take a closer look. A pluripotent stem cell has the potential to differentiate into any type of cell in the body. To become T-cells, they are put into a structure known as an artificial thymic organoid. This is a mini simplified thymus that is produced in the lab. Once these T-cells mature in the organoid, the chimeric antigen receptors are attached to the surface of these cells, and the cells are administered to the patient. In the end, with the allogeneic form of this therapy, it becomes cheaper and less time-intensive. Though much research is still going underway, CAR T proves to be a promising step in the right direction towards curing cancer, once and for all.